All right, everyone. I hope everyone had a good coffee and bio break. We'll continue with the rest of the program, and on the screen you can see a more detailed view on that. What we'll have is start with a mix of 4 young investigators, as I mentioned, starting with Wei Wu from Utrecht University, who will talk about the multi-omics human endocrine cell atlas. Wei, can you please take it away?
Hello, everyone. It's a pleasure for me to be here to speak to you about this nice project we have, which involves multi-omics analysis of human and enteroendocrine cells. And we made an atlas out of the RNA transcripts and secreted hormones from these cell types. In Star Trek, you have the United Federations of Planets. And in this EEC project, we founded an alliance with the goal of making new discoveries in gut hormonal regulation. And we formed a team involving proteomics and also organoid models, as well as transcriptomics. And the main questions that we wanted to ask were, what were the diversity in the human and EEC cell types, and also what are the gut hormones secreted in the human case. And we know quite a bit about the mouse models, but we also know that there are quite some differences. And most importantly, we are curious about the peptide hormone processing pathways involved in the human EECs. And these have strong implications in controlling diet and also the desire to eat and so on, and diabetes and many other important applications. And in this picture, you see an aircraft or spaceship that's labeled proteomics. It seems bigger in this picture just because of the standpoint I will take in this presentation. But of course, this is a very nicely integrated project involving multiple partners. So what we try to do here is to combine transcriptomics and organoid models and proteomics to study the EEC function in the human context. So from the transcriptomics, we will be able to decipher the transcripts made in single cells, and that allows us to subtype the EEC populations within organoid models that we can amplify these EECs with. And of course, from the proteomics point of view, we will be able to contribute in identifying the proteins that's expressed in these EEC subtypes, as well as the secretion of hormones. Some of these may be novel. And most importantly, we want to go after the peptide hormone processing pathways. Much of this is unknown. And the interesting part is that many of these hormones, when differentially processed, they have different biological functions. And as you will see in the next slide, the experimental model we use is the organoid system. And we know the mouse EECs have been very well studied. We have some idea of which hormones are dominantly secreted by mouse EEC subtypes. However, we do not know if this is true in the human case. So our collaborators in the group of Hans Clevers made these models made these models by overexpressing tagged versions of EEC peptide hormones, so that we can actually sort out by FACS the different cell types growing in organoids, and then we can study them separately in different populations. So as you can see here in the colorful image shown, that different EECs can be tagged by different colors, and in total we did for 6 known peptide hormones, and we see that the expression pattern of these hormones are quite different in different cell types, and also sometimes they overlay. In other words, that one particular EEC cell type can also express more than one type of hormones. And on the right, you see that we can well characterize the different hormones secreted. And also if we make organoids that's representative of the distal intestinal region or the proximal intestinal region, we can see specific differences in hormone secretion. And this is also very nicely validated by staining organoids that we can see the 6 different tagged versions of the hormones are well expressed. And on the right, you also see that by expressing and tagging these hormones in these EECs, it does not disturb the biological function of these EECs, that they still behave in an expected manner when triggered with a specific stimulus in terms of calcium response. And here I show you in panel A and B, you can see the single cell transcriptome data that can distinguish basically different kinds of EEC populations. And if you zoom into each of these EEC populations, we can then ask again, what is the expression of specific peptide hormones? And this is shown in panel C, that for instance, chromogranin A is quite abundant in EC cell population, for instance. And this allows us to look from a different view, that if we look at one particular hormone and how how widespread the expression of this hormone is in different hormone secreting cells. In panels D and E, you also see that we clearly validate that what we observe is specific to the human system, and therefore also affirms the importance of investigating these hormonal regulation in the context of human cells, because the behavior is really not the same if in the mouse model. So then what we, with these organoid models, what we went on to do was to isolate these EEC populations by effect sorting, that we can perform simultaneously the bulk RNA sequencing as well as intracellular proteomics, while from the supernatant that we collect from culturing these organoids, we can harvest the secretions from these EEC populations and to look further into the secretome. And particularly in the secretome, we are interested in 2 different samples. If we put the conditioned media from these isolated EEC cells through a molecular weight cutoff filter, we can separate the secreted products into 2 fractions. In the upper fraction, where you see this is basically retained in a 10 kilodalton filter, if we trypsinize that and put that on LC-MS, we can identify the full-length proteins that are secreted before hormone processing. Whereas in the flow-through fraction of this separation, we can inject that directly into the mass spectrometer for analysis of processed peptide hormones. And from this coupled analysis of the proteome, secretome, as well as bioactive peptides, we can see that at the RNA level, with transcripts, with single cell sequencing, we can actually cluster the different hormone secreting subtypes into distinct populations. And we can also do the same actually with the proteome profile of these hormone secreting cells. And interestingly, in the panel B, you can see that the expression, this is, I'm showing here the expression, the top 20 proteins that are most abundant in each of these subpopulations. You can see that the distribution of top 20 markers are also quite distinct, telling us again that these subtypes are different, really quite different, also intracellularly in terms of the proteins and the molecular function they perform. In total, we identified more than 2,000 proteins from very limited amount of cells, basically about 5,000 cells each of these fact-sorted populations. But what we also see is that while we identify more than 2,000 proteins from the total proteome, the secreted products we could identify from, for instance, the proximal EEC population and the distal EEC population are quite distinct from the total proteome. This gives us some confidence that what we identify are likely quite pure from the secretions and not including carryovers of cell products. And you can see highlighted here in yellow and in blue are the unique secreted proteins from the distal and the proximal EEC populations respectively. So because we analyze the secreted products in 2 different fractions, one which is a full-length protein product, whereas the other fraction includes the processed hormones, we can now start to correlate in terms of abundance or relative abundance within the analytes of the proximal and the distal EECs. What we can correlate is basically the level of proteins that are secreted and also how processed these proteins become in terms of peptides. And what you can see here, it might be quite small, but you see 4 different plots. On the top left, you have the EEC, proximal EECs, and you see that this particular protein, CCK, is quite low abundant, is identified in the almost the tail of the entire intensity distribution. But if you look at the processed hormone fractions, they see quite abundant peptide hormones. This tells us that we not only can profile from this series of experiments what proteins are secreted, but we also can tell which are the most dominant peptide hormones that are produced by these particular EEC subtypes. I show you here again, most of the proteins that we identify are well documented to be extracellular or secreted, giving us confidence that we are looking for real secreted factors from this in vitro model system. Through complicated analysis and also integrating this multi-level information, we actually made a secretome map of these EEC populations. And here I show you some of the peptide hormones and also secreted proteins that are specific to the proximal and the distal EEC populations. I indicated with purple arrows here that you can see there are some differences that are discorrelated between the secreted proteins and the processed peptides. This indicates an additional level of regulation that's happening at the stage of extracellular processing. Especially indicated, you can see the purple arrows here, chromogranin B, which is quite abundantly secreted at the protein level in the distal EECs, but almost never processed in the distal secretome, and so on. We also can derive a different kind of information. That is, if we look at a particular protein in panel B here, the top half shows you the processing map of motilin. And on the top portion, I show you here in color the non-bioactive peptides that are known to derive from this motilin full-length protein. And in the bottom panel, you see here all the processed peptides that's endogenous that we can actually detect from the organoid secretome. What you notice from the ghrelin processing in the bottom half is that we also detect new peptides that are stably retained in the secretome, whereas the biological function of these are yet to be discovered. I think most exciting of all is that maybe we could also find some new hormones that from this combined analysis with the RNA and proteome and secretome and bioactive peptide analysis. What you see here, I show you, is an mRNA transcript isoform 1 and 2 of chromogranin A. What is known about chromogranin A is that there are various exons, 1 to 8, that can be stitched together to give the full length chromogranin A, but also splicing of these and processing of this chromogranin A transcript can give various other peptide hormones, for instance, vasostatin 1 and 2, and other peptide hormones that are known to have very different biological functions. In this study, what we discovered was a separate mRNA isoform of chromogranin A that lacks the exon 6 that's likely lost in the at the transcript level of processing. But we could also detect the secretion of this peptide hormone in our experiment by reading off the amino acid sequence that links directly exons 5 and 7. And it really shows us that a new form of this hormone is secreted, lacking the exon 6. And collectively here with a very short presentation, I hope I've shown you some of the interesting data and inferences we derived based on compiling the evidence from human EECs at the RNA, proteome, secretome, and bioactive peptidome level. And I hope to show you the power of integrating this transcriptomic analysis together with proteome analysis of extracellular products that can actually not be visualized by using transcriptomics alone. And these exciting findings are published in Cell. If you're interested, you can look up the publication with the reference here. And this work is mainly done by the three first authors, Joep, Jens, and Julia. And this is a collaboration between myself and Hans Klevers.
Thank you very much, Wei. Very interesting talk. And again, I'll look up the paper for those interested. So thanks. The next speaker is Anthony Zucca from NanoString, who will elaborate more on their platform for spatial analysis of molecular biomolecules. Tony, please.
Yeah, it's not letting me. There we go. Great. Well, thank you all very much. I appreciate your time. So we're it's a little fried here with that transition. We're with NanoString, and we are really a gene expression technology company, and we have a couple different platforms. The nCounter, which is our bulk gene expression platform since about 2008, really robust with a lot of different sample types. And then about 2 years ago, we released the Genomics Digital Spatial Profiler. And this is the DSP Genomics.
This is this NanoString talk.
Yes, it is. This is NanoString. And so the geomics, the nCounter is the bulk gene expression technology that I mentioned, and it has quite a few panels for biomarkers, pathways, signatures. Like I mentioned, it works with quite a few different, it's very robust with different sample matrices. Works really well with COVID. But this presentation is going to be really more focusing on the Geomics DSP. And the DSP, I think I'm going to skip a couple of these. The DSP is what really launched the technology is this photocleavable linker. And this linker, when UV light hits it, releases a barcode. And this barcode can be read by either the nCounter or read by the NGS system, Illumina's NGS system. It's targeted, so it's either an mRNA target or an antibody. And then we mix it with either RNA scope morphology markers or fluorescent antibody morphology markers. The part of what the technology is a microscope, and that visualizes those morphology markers, show you the regions of interest, and then you define your regions of interest quite accurately. Then you shine UV light exactly where you want it, and then that UV light releases these probes. The probes get sipped up, and then the reading happens downstream with the nCounter or the NGS system. Very simplified. The real beauty is that you have a technology that can do whole transcriptome analysis all the way down to protein. Very simple in that there's very little image analysis. All the counting of these genes that are released by that UV light are done and brought back to the geomic system, and it's just genes, counts, and where they located on your tissue. We work with FFPE, fresh frozen. TMAs can be done very simply. We work with so many different tissue types. If you can do an ISH or you can do an IHC, that you can work on our technology. 25 plus publications so far and growing. We have quite a few targeted protein panels, about a 90 plex, can be customized, whether it's immuno-oncology or neuroscience and beyond. We have launched mRNA or RNA panels, an 84 plex, 1800 plex, 1,800 genes, and then 18,000 genes that are coming out. That'll be available by the end of the year, early next year. So one technology can do 18,000 whole transcriptome analysis and then bring it down to validation with protein. We've got quite a few different ways to visualize your ROIs. So with those morphology markers, and I thought I'd really show you a little bit here is one example in particular is a 44 plex by David Rim. And I think what this shows you the power, and it can be 44 plex protein or 18,000 with RNA. But what this visualizes is the regions of interest that were selected on those morphology markers. They illuminate these separately. And then inside these compartments, you can see CD56 expression in CD45 help for progression-free survival. Then another experiment done by David Rim that in melanoma shows that you can do it for cell populations. So here's melanocytes, and then you can UV illuminate these different melanocytes. And then you can also do that with CD68. And then once again, visualize the gene expression with that is being done inside those compartments, and an example of percent survival in CD68 areas. So I think I might have gone a little quickly because of the time constraints, but the technology has been really, really popular. We've been working with Dr. Edwin Copa at his powerful sequencing facility, and we're now having a collaboration with that group at the Utrecht Medical Center, and we're just excited to be continuing on. So thank you for the opportunity.
Well, thank you very much, Tony. Very interesting. And again, this is for very short times. It's only 5 minutes, so these are really pitches. But on the website, on the X-omics website, there's more information about different companies that provide their pitch here. So the next speaker is Bert Wouters from University of Leiden, talking more about analytical technologies for metabolomics. Bert, please. Yeah.
Okay, thank you, Alain, for the kind introduction. So indeed, my name is Bert Wouters, and today I would like to tell you a little bit about the research that we're doing in our group, considering a high-throughput flow NMR platform. I work in the group of Thomas Hankemeyer, one of the core members of the X-omics initiative. And the mission for our group, for the APSN group, is to accelerate the application of metabolomics in biomedical research, the clinical application of personalized health, and precision medicine. So to achieve our mission, we need high throughput technology, especially if you want to measure, let's say, tens of thousands of samples, 100,000 samples. You need automated high throughput analytical technology to enable this. And through development of this type of technology, we can then enable breakthroughs in metabolomic profiling, in life sciences, but also in other fields. In a similar vein, our group is, for instance, also working on the automated X-omics infrastructure in our group in Leiden, so that for X-omics starting next year, we can also start measuring tens of thousands samples for metabolomics. Now, if you look at analytical technology in general, there's always a compromise between your sample throughput and your metabolite coverage. If you would compare, for instance, direct infusion MS and LC-MS, with LC-MS you can reach a broader metabolite coverage, but at the expense of analysis time, and thus at the expense of throughput. In our group, we do a lot of mass spectrometry, but the topic or the project I want to talk about today is NMR based. We are collaborating with DSM in Delft to develop a novel sample handling platform for high throughput flow NMR. Both mass spec and NMR have, of course, their respective advantages, and I think they're quite complementary techniques. The advantages for NMR specifically are that it's a very quantitative technique. It allows for structural elucidation, and it's also quite robust. The downsides, or the bottlenecks, let's say, are that compared to MS, you need a larger sample volume, typically. Your metabolite coverage is a bit more limited, and NMR is not very sensitive. You're talking about micromolar concentration range. The goal for this project specifically was to focus on the throughput. So let's say for conventional 1D NMR, your throughput is 200 to 500 samples per day. And with a new approach, we wanted to triple this throughput. Now, in terms of increasing the throughput for this kind of screening NMR, what are the things we have to take into consideration? For screening NMR, we mean that we use a very short analysis time in the range of 30 seconds to 2 minutes. And typically this would occur with glass sample tubes. So one by one, the samples would be placed in the NMR probe in a glass sample tube. If your analysis time is quite short, then other steps in your workflow, the time for that, becomes very important. These time-consuming steps include, for instance, time needed for sample exchange, let's say 1 minute, time needed for temperature equilibration, up to 5 minutes, and also shimming, let's say up to 1 minute again. To tackle some of these time-consuming steps, to reduce the time needed here, we were inspired, of course, by literature, and we looked to an alternative for glass tubes, namely flow NMR. So here you use a liquid handling robot to move your sample through a stream towards a flow-through cell in the heart of your NMR system. If we look at different methods that are used for sample transfer in flow NMR, here in this slide you can see a bit of an overview of the conventional techniques. Conventionally, people would use, let's say, direct injection or flow injection analysis. And with direct injection, you completely fill your transfer tubing in the flow cell with your sample. Flow injection analysis is a bit more similar to liquid chromatography, where you have a carrier phase in which you inject your sample. In this case, the carrier solvent and the sample are both miscible. So the disadvantage here is that you get dispersion of your sample during the sample transfer step, which leads to a lower concentration of your molecules in your flow cell, which hampers your sensitivity. With direct injection, you do reach a sensitivity comparable to glass tubes, but here, for instance, you have to incorporate extensive rinsing steps in between the samples, which hampers your throughput. An alternative that has been used is segmented flow with air bubbles. So here you use air bubbles to separate your sample segments in the transfer tubing in the flow cell, which has the advantage that there's no dispersion of your sample. However, disadvantages here are that you still need to carry out rinsing steps because your sample is still in contact with your flow cell, and that also the air bubbles, because of their magnetic susceptibility difference, can hamper the quality of your NMR spectrum. So what we propose is the fourth option in this slide, is using segmented slug flow with a fluorinated oil instead of air bubbles to separate the samples. The main advantages here is that we use a fluoropolymer surface for a flow cell in combination with this fluorinated oil, which creates a preferential wetting effect. So there will be an oil film present on the inside of the flow cell, which greatly limits the carryover effect. Additionally, we also have magnetic susceptibility matching between the oil and the water sample, which is also of benefit for the quality of your NMR spectrum. So this is a bit of an overview of what our approach looks like. On the left, you can see a liquid handling robot. This is a CTC PAL. Here in a temperature-controlled sample hotel, we can store our samples in 96 well plates. Then with a syringe tool, we can move a certain sample volume towards a switching valve with an external loop. With this switching valve in this loop, we can create this segmented flow. So we have up to 3 meters of transfer tubing between the robot and the NMR flow cell, made from a inert fluoropolymer material. And if you look at the zoom in the middle, you can see that here then we have the immiscible carrier fluid, the fluorinated oil, and an isolated sample slug, which is not in contact with the surface of the tubing. On the right, you can see the NMR with at the heart the flow cell. Once the sample arrives in the flow cell, we can park it there by stopping the flow, after which we perform our analysis, let's say between 30 seconds and 2 minutes. Once the analysis is done, the sample can be transferred to waste, and the next sample will get in line. Now, to develop this platform, we had a couple of important aspects to take into account, 4 of which you can see here. And during the next slides, I will start discussing these aspects one by one. The first aspect, of course, is the approach we take for sample transfer. Here we take segmented slug flow as our approach. You can see in the diagram a bit of a visual representation. And the most important thing here, as I mentioned, is that because of the immiscibility between the 2 phases, you have sample transfer with 0 dispersion. So your sample phase behaves as a discrete microreactor. The exact morphology of these phases, whether it's droplets or slugs, depends on a very complex interplay between physical forces. So you have viscous forces, gravitational forces, inertial forces, and interfacial forces. In our approach, we use this for sample transfer, but if you look in literature, segmented flow has also been used for sample storage, for mixing of reagents, and for high throughput analysis. During the development phase of our project, we also developed a non-intrusive laser sensor to characterize our samples. So to at the transfer tubing, we incorporated a laser sensor of 660 nanometers. And the laser beam in this case was perpendicular to our transfer tubing. So here horizontally, you see the transparent fluoropolymer transfer tubing through which your sample is flowing. And then by different amounts of light that end up at the receiver, we could distinguish between the carrier phase and our sample segments. We also had differences here in, let's say, the refraction of the light, the scattering of the light, differences in absorption. So as you can see here at B, this laser sensor provided us with a very clear, almost binary signal, which also allowed us to identify up to 10 different solvents. An additional feature of this laser sensor is that we could also assess the quality of our sample slugs. So ideally your sample slug is intact, but in some cases there can also be fragmentation of your slug. So here in this image you can see that this laser sensor also provided that kind of quality control for our samples. After the sample transfer to the tubing, we'll of course end up at the flow cell. At this point, we were confronted by some limitations of the commercial existing technology. Typically, here you work with a glass flow cell, which is fragile and quite expensive. And it's also not an ideal combination with segmented flow with fluorinated oil. And this has to do with the preferential wetting effect, for which you need a fluorinated surface. Additionally, as a disadvantage of this glass flow cell is that the connections for the flow cell were also limiting us in terms of back pressure and flow rate. So it did not allow us to reach very fast sample transfer times. For this reason, we started redesigning the flow cell, and we opted to make our own flow cells from a permanently fluorinated surface material. So in this case, we used PCTFE, also known as CalF. And this permanently fluorinated surface allows you to reach a very low carryover between your samples. And as you can see here at the top, on the left-hand side, you see the end of the Bruker cryofrit accessory, which is used to lower the flow cell in the NMR. Our flow cell is fully compatible with this technology. So you can simply exchange a fluoropolymer flow cell with an existing glass flow cell without any adjustments. And finally, what I want to point out here is that we also have these threaded ferrule connections in our flow cell to connect the transfer tubing to the cell, which allows us to use a higher pressure and thus higher flow rates to enable sub-30-second transfer time. We've been working together now for about 1 year with our partners at DSM. And the main take-home message for this slide is that if we compare our glass flow cell, our fluoropolymer flow cell with a glass flow cell, is that we have a 3 times lower carryover between samples, but at the moment we still have the challenge of slightly broader peaks. So this is still an ongoing challenge that we need to address. Alain is reminding me of the time, so I'm quickly just gonna go to the last important slide. After trying out the standard samples, such as sucrose and maleic acid, we've also, of course, done a number of multi-component samples. Here's an example for non-alcoholic wine. And here you can see that our results are very comparable to typical NMR results with glass flow cells, and that in just 8 scans, or about 20 seconds, we can identify up to 18 metabolites in wine. Finally, for the last couple of seconds, I would like to thank the partners for this project. So especially Rafael at the Fine Mechanical Department and Paul Michiels in the ABS group for all the results that I was able to share with you today. So, thank you.
Thank you very much, Bert. Again, a different talk, which is very interesting. I really like this mix which we're having during the day. So thanks for your contribution, Bert. So we're now switching to the contribution from Bruker, and Gary Krupa will actually replace Robin Tuku, who was originally planned to speak. And Gary will talk about spatial omics, how spatial temporal cross-omics can improve our understanding of tumor microenvironments. Okay, Gary, it's a very short, literally a pitch. But again, I'd like to refer all the participants to the website of the cross-omics website, where all the links are there. So thanks, Gary. Next speaker is Hans Wessels from the Radboudumc, close to home. Hans, please take it away.
Thank you, Alain, and welcome everybody. So there are millions of distinct unique protein configurations present in the cell that we typically refer to as proteoforms. And these proteoforms come about via unique combinations of both the amino acid sequence and the post-translational modifications that can be present. In our lab, we have focused on protein glycosylation already for quite some time. And we have a particular interest into this carbohydrate that's attached to protein structures. So why is that? Well, our lab operates in a clinical environment, and many of the diseases that we look at, we tend to use the protein, well, in blood or plasma for diagnosis, to monitor disease, and also preferably to understand disease. And when we look in blood or in plasma, more than 90% of all proteins happen to be glycosylated. And this glycosylation actually is a major contributor to the diversity of proteins that we have in the human body. On a single site, there might be tens of different glycan structures that can be present, thereby multiplying the diversity that we have at the protein level. Glycosylation is not just a modification that decorates the protein, it's actually there to modulate its protein biology. And we know from several glycomics studies that have been performed in the past that glycosylation indeed changes in many human diseases, both genetic and acquired. However, protein glycosylation is still rather unexplored at the site-specific modification level. So we decided that this would be a good opportunity to look if we could use protein glycosylation and glycoproteomics for improved personalized diagnostics. So just a brief overview of the workflow that we perform. We start off with a plasma sample that contains proteins and glycoproteins. We then cut these proteins into smaller fragments using trypsin, so they are better usable for LC-MS analysis. We then specifically try to purify or enrich our glycopeptides, which are then measured by liquid chromatography combined with tandem mass spectrometry. In this case, we try to separate our glycopeptides using reverse phase chromatography, and as soon as the glycopeptides leave the column, we ionize them, so we put a charge on them so that we can analyze them as intact glycopeptide, looking at the mass and the intensity, but also fragmenting them in two different ways. First of all, we fragment them at low collision energies so that we can sequence the glycan, so we get an idea of what the glycan composition and sequence is. And we perform collision energy, elevated collision energies, so that we actually get some sequencing information from the peptide moiety, so that we don't only know what the glycan looks like, but also what it's attached to. Once we have that information, we can link that to all the signals that have been acquired for independent samples. So on the left here, we have all the independent MS data files. That means that we recorded signals for all different samples that have been measured. We then combine all this information from all the individual measurements into a single consensus feature map. So a feature in this case is just a unique distinctable LC-MS signal. And then we take our identification data and map that on it and continue to biomarker discovery and glycobiology analysis. And this is a rough view of what the raw LC-MS data would look like. So on the x-axis we have retention time, and on the y-axis we have our typical master charge ratio. And essentially, each of the dots that you see here corresponds with a unique LC-MS feature. And using our identification data on top, we can reconstruct the amino acid sequence and look at the glycan sequence where possible. So when we analyze the plasma proteome, what do we get back? Well, essentially we see the glycosylation status of many different proteins in plasma. And the nice thing about this is that we can now follow the glycosylation actually site-specific manner. And we see that for many different proteins in one single experiment. When we look at the data, that shows us that we see 3 distinct clusters of protein glycosylation profiles to be present. The red cluster, which consists of the well-known complex N glycans. In blue, we have shorter glycan fragments that carry fucose moieties. And over here, we have an isolated cluster of some high mannose glycosylation species. And when we look at a single protein like complement component 4B over here, we can see that one side can carry actually the complex N glycans, whereas the other side might well be part of this green cluster over here that carries the complex high mannose structures. So how to evaluate this, whether it would be useful for clinical applications? Well, for this we used samples from patients that suffer from congenital disorders of glycosylation as a model to evaluate the overall performance, but also clinical relevance. First, what are N glycans? N glycans are heterogeneous carbohydrate structures with a common GLUC-NAC mannose 3 core that's visualized over here, and that are linked to the asparagine amino acid residue of proteins. What is rather unique for N glycosylation in comparison to other PTMs is that it's a multi-organellar synthesis process that starts off in the ER, and via trafficking it gets transported to the Golgi, where further N glycan synthesis continues until we have a finalized process. What's also important to know is that this full glycan synthesis process is not template driven, so it's an enzymatic process that takes place. And overall, we hope that protein-specific N glycosylation, if we can follow that, then we get a much better idea of what's happening in terms of glycobiology, but at the same time, also improve diagnostics. So I'm skipping quite a bit of work down to this analysis. And what we performed here is that we took binary comparisons between patients with a particular CDG gene defect, 10 of them listed here, and performed partial least squares discriminant analysis versus set of 40 control samples. And when we look at the analysis results, we see that for all 10 CDG defects, we get a black and white classification result, which means that every patient is classified as patient and every control sample is control. When we compare that to the results that we obtained with intact transferrin mass spectrometry, we see that for 4 out of the 10 CDG defects, we get a black and white classification result. So in some more detail down below here for nonce, first on top, we have a principal component analysis. So we have the score plot, and we can see the 40 green control samples and the non-samples in orange, and you can see that by themselves, using an unsupervised approach, they cannot immediately be discerned using transferrin, intact transferrin data. Whereas when we look at the glycopeptide data without doing any statistics or anything on that, we already get a clear separation. This is also reflected by the PLSTA performance. So on the left, we have the classification results for healthy individuals, and to the right, we have the classification results for our patients. So for transferrin, we see over here that there's a misclassification of some patient samples, whereas for glycopeptides, it's a clear-cut separation, both for nons and COF5. So this will actually enable us, has the potential to enable new diagnostics in CDG patient care. Binary comparisons are only the beginning of this, of course, and we were also interested if we could classify samples from patients according to specific gene defects. And in order to do so, we used a supervised learning or genetic algorithm random forest approach. And on the left here, in the rows, we have our patient samples or class samples, and on the right, we have the classes that the algorithm will try to classify the samples to. When we look at that, when in green, we can see that all the 40 controls have been classified as control. And overall, for 104 out of the 108 samples, we have correct classification down to the gene defect. Team M199 is particularly interesting because this defect, one patient has been classified as an HB6AP1 and 2 patients as an HB6V082. But this is kind of logical because both Team M199 and the ATP defects all lead to a VHPase enzyme deficiency. So whether or not we could discern that at the genetic level would be a rather big question. So according to Wessels, the only true misclassification in this case is the classification of CCDC to PGM1. When we look at the features that are used to make this classification model, and we analyze them in a non-supervised way using t-statistic neighbor embedding, we can clearly see clusters of different patient samples clustered together. When we look at these in a little bit more detail, interestingly, we can see that we have most of or all the healthy individuals over here on the top left. Over here, we have close vicinity clustering of different culture homeostasis and trafficking defects, some isolated enzyme steps, and down below we have sugar metabolism defects. So apparently, the data contains information that allows us to also look at what is similar between the different defects, and in this case it nicely correlates to the disturbed functional process in the cell. Another question that we wanted to answer is whether or not the glycosylation changes are also clinically relevant. And for the sake of time, I will go through one of them in this case. So over here we have fucose transporter deficiency, which means that we would expect that fucosylated glycans would not be made anymore or reduced. And when we look over all the different glycosylation sites that we measured, we see a clear reduction in fucosylated glycan species, which reflected in the panel on the top right, where we show that all these fucosylated species have been lowered in patient versus control, whereas fucose lacking species, so the glycans without fucose, are actually increased. We did that for multiple genetic defects, and we can see that it nicely follows the exact steps that we would expect it to be. So although we didn't show it today, we also know that tested the analytical robustness and performance of the technique. And we were happy because we could now take this further into clinical implementation. But in order to do so, there's several things that we should consider. First of all, it's rather impractical at this point to include several 100,000 different signals in routine diagnostics. And translating that into an easy to interpret, unambiguous report is a rather challenging case by itself. In the end, we also need to make sure that complete workflows need to be compliant with ISO guidelines, of course. So the classical way of how biomarker discovery and implementation handles is that we first measure a patient cohort using holistic measurements. That means that we record as many signals as we can for a single sample, do our statistics, identify biomarkers, develop a targeted assay, validate the targeted assay, and then finally implement it into clinical care, where we take our patient sample, measure only the biomarkers of interest, and then in the end report and hopefully get to diagnosis. For efficiency reasons and retrospective analysis, we rather went for this particular route where we analyzed the patient sample using untargeted holistic data acquisition. So essentially we report every possible signal that we can measure for a patient, which is still perfectly fine to use for biomarker discovery and research purposes. But at the same time, for the diagnostics, we can simply extract a list of targets or biomarkers in a targeted way so that from the holistic data, we get our targeted data, which we then translate into a glycobiology report for diagnostics. So how does it look like? Again, over here, we have the raw data with lots of signals. And in this case, we only extract these features of interest for 2 different immunoglobulins and 2 glycosylation sites of transferrin. So that's much easier to handle computationally wise. So what does it look like in this case? So again, we have the fucose transporter deficiency. Over here, the black dots are for the control samples. 2 green dots are 2 independent pools of 5 random healthy donors that we use as batch control during measurements. And over here, we plotted the non-fucosylated glycan fraction on IgG1 and IgG2. And here we can see the 2 different time points from the same patient in red, clearly distinguishable from controls, whereas there's a clear reduction next to it for the fucosylated species. So although we measured over 40 different glycans, in these cases for 2 proteins, we can still summarize it as something that can be evaluated directly by a clinical expert. Another case example here would be monosidase deficiency or MAM1B1 CDG. And over here we have 2 different glycosylation sites, so transferrin at asparagine 432 and at position 630 of the canonical sequence. What we see is that our patient has a clear reduction in complex glycan types and an increase of hybrid glycans. We only see that at asparagine 432. We don't see it at the other glycosylation site. When we look down below, then we can see that this is mainly caused by the expression or the availability of a diagnostic hybrid glycan, which is really specific for our patient. So this is rather straightforward to interpret. So I'm going to wrap this up, but so far we know that glycoproteomics can be used to get better insights into protein glycobiology at the site-specific level, and we now look forward to implement this technology first in CDG diagnostics, but also other diseases. And I would like to thank our colleagues from Radboudumc University, Bruker, and BSI for their help and support of this project. And in the end, I would also like to give out a call for a manuscript for a special issue on glycoproteomics. And with that, I would like to thank you for your attention, and I hope you will enjoy the festival.
Thank you, Hans. So this invitation is well seen. I'll leave it on the screen for a bit. And then we move to the next speaker, which is Ruben from Microsoft. And invited Ruben also to join our festival, also to elaborate more on the data platforms that Microsoft has. So Ruben, the floor is yours.
If I can be heard, I'll gladly kick off. Thank you for the opportunity. My name is Ruben Pol. I'm a cloud solution architect at Microsoft, helping our customers to become familiar with the tools that we have to offer in terms of cloud. And in that capability, I've been also working with Alain for the Radboudumc, setting up the digital research environment there. We have done projects at the Erasmus MC in genomics, but also have worked on high-speed genomics pipelines at agricultural partners, for instance, like Keygen, really making their processes more efficient. Let's see, I can control this. So next slide. Let me.
Yeah, there's a slight delay, Ruben. So
okay.
It's hard, but we need to be patient.
Yeah, but I'm pressing it now. And I'm not of problems, right? The heavy compute workloads, so large jobs that need to be done, large pipelines that need to be run, where we can attach scalable compute to those to that storage medium. And of course, what we hear a lot, what I have heard from many researchers, is that they just want the flexibility to work with new tools and technology, and definitely not only Microsoft technology, but also open source and in the breadth of the platform. So a self-service platform with open source tools is what they'd like to have, where of course cloud can also help to quickly attach new tools and deploy new tools on demand. But when we're moving to the field of cross-omics, there are some added challenges, or the challenges become even more pronounced, because we're working with a heterogeneous data landscape where we want to combine research fields with, of course, different standards. And one issue there is data management in that heterogeneous data landscape, where we help to offer a standardized data platform, so use the same fundamentals in terms of security and operability. But of course, there's a need also for processes and skilled data stewards to support that. Second of all, the compute workloads become even more unpredictable and spiky. So when combining data sets, they can rise exponentially, these needs. Maybe also with specific compute, like not only CPUs, but also GPUs or programmable microchips that can be offered through cloud computing, as well as an extensive container platform. And finally, in terms of data science, we want a collaborative platform where we can do that in a reproducible way. So a shared data science platform, people from different teams can work on the same problem. And we've learned a lot from we are using a lot of our knowledge from our DevOps experience in Microsoft. So there's a bunch of tools that accompany that. I won't go into them, but they are here in the slides for future reference. But what I would like to do is show them to you in an architecture, how this works together, and only take a minute or two. So what you have here on the left side are basically all the different data sources of on-prem systems. So for instance, the genomics data, but also the tabular data, media files, etc. And our data factory service connects to these on-prem systems, so it creates a trusted link to those, so you can synchronize or migrate, if you will, and moves it onward to our different forms of storage. So we have a data lake form of storage, which is basically a cheap, scalable way of storing unstructured and semi-structured files like genomes, but also documents and media files. And we have our SQL technology, which is more the traditional database technology, where you have a very just highly capable way of storing data. For instance, if you're looking at metabolomics cohort information or other tabular data sets that may be also very large in scale. We have a way of exploring that data, searching it and mapping it. We have specific sequencing tools in our own Microsoft Genomics service, but also code first in our Databricks service, where you can scale it up all the way to map an entire human genome within the hour. And then we have our Azure machine learning environment, where you use tools still using Jupyter notebooks, so using Python and R languages to map experiments in there. Finally, we also have one of our new services, which is Azure Synapse Analytics. That's a combination of the modern data warehouse, where you can combine the power of Azure SQL as a MPP platform with Hadoop Spark. So using both in-memory and SQL-based scalable compute. Then send that onward, of course, to, well, we have our own Power BI service, but you might prefer the ways of consuming and visualizing that you are used to. So Jupyter notebooks or libraries in those languages, but also custom applications that can be served from that data platform. All of which is backed up by basically our standardized cloud security tools, blueprints to basically store all this infrastructure as code. So we'll be able to redeploy everything. And of course, the tools to do an efficient cost management and overview of all these services. With that, I move to the last slide and I thank you. Important to note is that you can, as this image shows, you can use a lot of your open source tools and embed it in this platform, and how that works and how that integrates. We are very interested in helping you setting up a proof of concept for your cross-omics environment in the cloud to make this a bit more concrete. So with that, I wrap up. I thank you for your patience, especially during the dropout. I hope this was interesting and relevant to you.
It certainly was, Ruben. Thank you very much. And this block is Jenny van Dongen from the Free University in Amsterdam, our first year of today, talking about DNA methylation signatures. Please, Jenny.
Yes, thank you very much. So I will talk about our epigenome-wide association study of educational attainment that was performed in 4 Dutch cohorts.
Here we have the same issue as we had a moment ago, Jenny.
It seems so, yeah.
Yeah.
So this work was done within the BIOS consortium, which was initiated within BBMRI-NL. And the goal was to facilitate a large-scale infrastructure to analyze genetic, metallome, and transcriptome data. And this included 6 Dutch biobanks, approximately 4,000 individuals for whom genetic data and rich phenotype data was already available. And these samples were then measured together on the Illumina 450K array to generate metallome data. And RNA sequencing was applied to measure the transcriptome. You can have a look at their Google Scholar page to see the full list of publications from the BIOS consortium. And there are 2 links on my slides that you can have a look at if you would like to see how to gain access to the BIOS data and what other data is available within BBMRI-NL, which includes many more biobanks and also, for instance, metabolomics data. So within the current study, we analyzed data from 4 cohorts from BIOS, which are the large population-based cohorts. And we mainly focused on peripheral whole blood DNA methylation data. So we first performed an epigenome-wide association study in each of the individual cohorts, and then combined the results in a meta-analysis. And in the table, you can see some of the cohort characteristics, and all the results that I will present today have been previously published in the Nature Partner Journal Science of Learning. First of all, we harmonized the educational attainment data that were collected within these 4 cohorts. So we defined educational attainment as the highest completed level of education at the age of 25 or higher, according to 7 categories. So the first one is only primary school, and the highest level of education was a university degree. And this slide shows you the interesting trend of educational attainment in the Netherlands across birth cohorts. So we have the 4 different BIOS cohorts represented by a different color box plot. On the horizontal axis is the birth cohort of the participants of these cohorts, ranging from 1925 till 1989. And on the y-axis is the educational attainment. So during this period, the compulsory years of education has changed within the Netherlands from just 6 years in 1901 till at least 11 years currently. And you can see that there is a clear increase in trend. And this trend was sex-specific, so especially for women in older birth cohorts, it was just very rare to obtain a higher degree of education. So we took these trends into account by transforming the data to a RIDDIT score, which reflects the level of education of a person relative to other individuals of the same sex and born within the same birth cohort. So this transformed score was then used in our epigenome-wide association study, in which we simply tested if there is a linear relationship between methylation level and education level for approximately 450,000 methylation sites in the genome. We adjusted for several commonly used covariates such as age, sex, white blood cell counts, and technical covariates. And in additional analyses, we looked at the effect of additionally correcting for smoking status. Educational attainment is a very interesting trait for several reasons. It is an important correlate of several cognitive and personality characteristics, and the advantage of educational attainment is that it's very well documented, while, for instance, detailed IQ tests are not as readily available. But it's also a very strong indicator of the social environment of a person, and it's very strongly related to health differences. For instance, if you look at the average life expectancy at birth, which is approximately 79 in Dutch men and approximately 83 in Dutch women, there's still a very large difference between higher and lower educated groups. For instance, higher educated men on average live 6.5 years longer than lower educated men. So by identifying loci in the genome where methylation level correlates with educational attainment, we may potentially get insight into the loci whose epigenetic regulation is involved in or is a biomarker for differences in cognition, social environment, and also educated-related health differences. So I would like to show you some of our results. First of all, the results from our meta-analysis where we did not yet correct for smoking. This plot shows p-values at all different locations in the genome, and we identified 58 sites whose methylation level correlates with educational attainment. I'm just going to point out a couple of interesting genes. The CNTNAP2 gene is a target for the transcription factor FOXP2, which is very well known for its role in language development. And genetic variants within CNTNAP2 have been associated with several neurodevelopmental phenotypes. PCDH9, it's a gene of which we know that copy number variants within the gene have been associated with autism spectrum disorder. It's also been studied in mouse models where similarly knockout of this gene has an effect on social and object recognition. It's also been described as a tumor repressor gene. Our top CPG is located in the arrow hydrocarbon receptor repressor, and this CPG is actually very well known from epigenome-wide association studies because it's one of the most strongly associated and best replicating hits for various smoking phenotypes. In fact, all of our 58 CPGs have been previously associated with smoking, and 50% have been previously associated with prenatal maternal smoking. So we know that smoking is more common among lower educated people, especially in the later birth cohorts. So we looked at the effect of correcting for smoking status. This was our initial Manhattan plot, and when we adjusted for smoking status, the effect size of most CPGs was reduced to various extents, and 11 CPGs were still genome-wide significant. So we then looked whether the remaining association between these CPGs and educational attainment could be explained by differences in how much people smoke and for how long they have smoked. We looked at this in data from the NanoString register. I will just show you the results of our top CPG in the AHRR gene. So this is the effect size for the relationship between educational attainment and methylation level, not adjusting for smoking. Then this is adjusted for smoking status, so the effect size is reduced. And it's a little bit further reduced if we also adjust for smoking pack years, but still we see that there's a remaining association with educational attainment, even after adjusting for smoking status and pack years. Adjusting for maternal prenatal smoking did not have a very large effect in this adult population. And if we repeated the analysis in never smokers only, the effect size was greatly reduced, but we still saw an indication of a remaining association between methylation level and educational attainment. And again, adjusting for maternal smoking did not greatly change this association. So I'd like to show you a little bit more about the pathway in which our top gene is located. So the aryl hydrocarbon receptor is a receptor for various environmental toxins, including dioxin and PCBs, some of which are also present in cigarette smoke. And this receptor is a transcription factor that regulates a number of genes, not only genes involved in xenobiotic metabolism, but also lots of developmental genes. And it also regulates its own repressor. So this is a negative feedback loop. And based on animal studies, it is known that this pathway also plays a role in the developing brain. So if mice are exposed to dioxin, which activates this pathway, if they are exposed prenatally or lactationally, this actually can disturb neural network formation in the brain. Of course, we analyzed methylation data from blood within the BIOS consortium because we did not have access to brain methylation data. But we looked at several publicly available data sets to get some indication of what these methylation sites, how they behave within brain tissue. So first of all, we looked at a data set of fetal brain methylation data. And we found that 18 of our 58 CPGs show dynamic methylation level in the fetal brain. So that means that their methylation level changes with age within the brain of the fetus. And here are just a couple of examples. The first plot shows the CPG with the strongest relationship between fetal age, which is on the x-axis, and methylation site on the y-axis, and the second CPG is our top sites from the EWAS of educational attainments. A second data set included methylation level from matched blood and brain samples from post-mortem tissues. And based on this data set, we could look up the correlation between blood and brain in adults, and we found that 10 of our 58 top CPGs actually show a correlation between methylation in blood and brain, indicating that to some extent the variation that we see in blood is also reflective of variation between individuals in the brain. So to get a little bit more insight into the types of environmental exposures that correlate at the epigenetic level with educational attainment, we obtained results from previous EWASs of several different environmental exposures. We took the significant CPGs from those analyses and computed the correlation between their effect size for the exposure and the effect size of the same CPG in our EWAS of educational attainment. And this showed, first of all, that there's a strong correlation for smoking. There was also a correlation for prenatal maternal smoking, and this is especially the case for the CPGs that still show a methylation difference in older children, so not just in cord blood, but where the effect of maternal smoking persists in older children. But there were also correlations with other exposures. So this is prenatal maternal plasma folate, where we saw a small correlation that was positive, indicating that higher educated people are exposed to higher folate levels. And there was also a small positive correlation for fine matter air pollution. And that could reflect that within the Netherlands, higher educated people more often live within cities where there is more air pollution. There was no significant correlation with alcohol consumption, although we did find that several of our set of 58 CPGs have also been previously significantly associated to alcohol consumption. So we also looked at genetic influences on these methylation sites. We obtained data that we had previously published based on a twin study and a study where we also combined methylation data with genome-wide SNP data. And we found that yeah, these CPGs do not seem to be solely influenced by the environment, because their average heritability was 58%, and that's shown in this graph. So the blue bars show the twin-based heritability for these 58 CPGs, and the red bars show the SNP heritability, so the variance in methylation that is explained by all genome-wide SNPs. And on average, 19% of the variation in DNA methylation at these educational attainment associated CPGs was explained by genome-wide SNPs. So it seems that not only the environment, but also genetic variation contributes to differences in methylation at these locations. And with that, I would like to conclude my presentation. So we know that educational attainment is very closely related to health, also to Alzheimer's disease and societal inequality. So we think that our findings have yeah, important implications probably for these health differences. But bear in mind that cognition yeah, mostly takes place in the brain, while we have methylation level measured in blood, so that limits the conclusions that we can draw. We did find yeah, indications that the methylome that we measure in blood can give yeah, an important insight into the environmental differences to which higher and lower educated people are exposed. And with that, I'd like to thank all co-authors on this project, and especially the BIOS Consortium and BBMRI-NL. And the results of this study have been published on the BBMRI-NL Atlas. And you can also access all quality control and analysis pipelines that were used for analyzing the methylation array data. Thank you very much.
Right, thank you, Jenny. So with that, we're closing this second block in our symposium.